Right, so I'm really delighted to give a presentation on the clinical trial process today. So clinical trials are part of the drug development process, which is summarized on this slide. And I will go through this slide, starting on the left by the first step, which is the drug discovery stage. It's very important to understand the biological mechanism that underlie the disease in order to identify targets against which drug can act and reverse the symptoms of the disease. So here in this talk, when I'm going to talk about drug, I mean any substance that has a physiological effect when it's introduced in the body. So it can be a gene or cell therapy product or a pharmacological agent. So once compounds against targets have been identified, they move through the next phase, which is called the preclinical stage. That happens also usually in a laboratory, in an academic lab. And this is a step where the drugs are being tested in disease models, usually it's animal models, before they are being tested in humans. And so here, the goal of the preclinical stage is really to assess the safety of the drug as well as the efficacy. If this is successful, the sponsor is going to submit what is called the investigational new drug application to the regulatory agency of the country in which it wants to test the drug. So, for example, it's the Food and Drug Administration, FDA in the US, or it is called the European Medicine Agencies in Europe, or other names in other countries. So the regulatory agencies is going to give the green light in order for the drug to be tested in humans in what is called clinical trials. And clinical trials are composed of three phases: phase 1, phase 2, and phase 3. The first phase, phase 1 clinical trial, the goal here is to determine what is the dose of the drugs that is going to be tested that is safe. Usually this phase is testing the drug for a short duration on a very small number of patient population, about 10 in rare diseases. If this step is successful, then the drug moves to phase 2 clinical trials, where here the drug is going to be tested in a larger sample patient population, about 10 to 20 patients. And here the goal is to assess the safety of the drug, but also to start collecting data about the efficacy of the drug. If that phase is successful, then the drug can move to the next phase, which is the phase 3 clinical trials, where the drug is going to be tested for a longer period. Usually it's about 1 year in a larger sample of the patient population. So for rare diseases, it's about 100 patients. And here, in addition to the safety of the drug, what is really important is to gather data about the efficacy of the drug. If this phase is successful, then a new drug application is going to be submitted to the regulatory agency, who is going, based on this data, to give a market authorization for the drug. So the drug development process is not finished at this stage. There is what is called the post-marketing period. So this is the time during which discussion with insurance companies are taking place, which may ask additional studies, which may restrict the reimbursement of drugs to a subpopulation of the patient population. This is also a time where the drug price is going to be discussed. And during that period, and for an indefinite period of time, the drug is going to be monitored in the real world to also assess, the long-term effect of the drug. So all this happens before the drug is accessible to the patients. So this drug development is long. It takes about 10 to 15 years to develop a new drug. It is very costly. It's estimated for common disease to cost about $2.6 billion to develop a new drug. It's a little bit less for a drug for rare disorders. And it's a very inefficient process, since 55 to 90% of drugs that enter phase 1 clinical trial will fail. So here on this slide, I would like to spend a little bit of time explaining some important concepts that are used in clinical trials. So the first one is randomized placebo-controlled clinical trial. So this is really the gold standard of clinical trial. This is a trial that has been designed well enough so as to be able to distinguish the effect of the drug from other influences, such as the placebo effect. So what is a placebo? It's an inactive drug that is indistinguishable from the drug that is used in a clinical trial. By opposition, an open label is a trial in which no blinding is used, so all the parties are aware of the treatment that is being administered. Usually it's the drug. And very often, a randomized placebo-controlled trial is going to be followed by an open label phase, so that it gives the opportunity for all participants of the clinical trials to receive the drug. So let's take an example as shown on this graph here on the right-hand side. So this represents a sample population that is supposed to represent the patient population. In a randomly fashion, a subgroup of the sample population is going to receive the drug. This is the drug treatment arm, whereas another sample randomly is going to be attributed the placebo. This is the placebo arm. So what are we going to measure in a clinical trial? The most important is what is called the primary endpoint. This is the primary outcome measured by a clinical trial to assess the effect of a drug. Primary endpoints are required by approval by regulatory agency. In fact, it's really based on these primary endpoints that the regulatory agencies is going to give or not a market authorization for the drug. There are also secondary endpoints. These are more exploratory endpoints. These are measures that may support the primary endpoints or demonstrate additional clinical effects of the drug. So let's take an example with this graph on the right-hand side. So on the y-axis is represented the level of hyperphagia, and in function of the time in clinical trials. So during the first phase, which is called the baseline, no group is receiving any treatment. So the level of hyperphagia should be similar during that phase. In the placebo group, ideally, the level of hyperphagia shouldn't change compared to the baseline. Whereas if the drug is efficacious, it should decrease the level of hyperphagia, as indicated here in the yellow line. And the drug effect is really the difference between the difference of the level of hyperphagia between the drug-treated arm versus the placebo arm. So there are two key messages that I would like to add here. The first one is that it is really important to test a new drug within a clinical trial framework under the monitoring of regulatory agencies who are taking every safeguards at every step to avoid any toxicity of the drug in patients. But nevertheless, this can happen. And one example in Prader-Willi syndrome is the beloranib trial by Zafchen, who had really a very nice effect on hyperphagia, but unfortunately led to two fatal cases due to pulmonary embolism and deep vein thrombosis. And the trial was outed. The other message that I think I really want to really stress is the importance of the placebo group. And I would like to show you two examples. So this is one example that has been extracted from a recent publication. So on the y-axis, you can see here a little bit different. It shows the hyperphagia change from baseline. So if this change is in the negative range, it means that the treatment has decreased the hyperphagia. If it is in the positive range, it means the hyperphagia is increased. So based on these slides, you can see that here the drug seems to have a significant effect in decreasing the level of hyperphagia. But then if you look at the placebo group, you can see it has also an effect, meaning that in the placebo group, you see a significant also decrease of the level of hyperphagia. And then if you measure the change between the placebo arm and the drug arm, there is no statistical difference. So here the drug doesn't have an effect on the level of hyperphagia. So on this slide is another example that shows you the level of hyperphagia in the y-axis in function of the clinical trials duration. And you can see here in the blue group, it looks like there's no effect of the treatment on hyperphagia, whereas there is a significant decrease of hyperphagia in this group indicated in orange. But contrary to what you may think, in fact, there is no effect in the drug-treated arm, whereas there is a significant effect of the placebo arm. So with this, I'm going to stop, and I would like to thank you, and I'm happy to take any question at the end of this session. Thank you.